Okay, good morning everybody. This is now going to be the recording for, ladies and gentlemen, please. This is the recording for the second part of UROCHORDATA and CEPHALOCHORDATA for your lectures. Remember we did UROCHORDATA last week. The Ascidians, all of those different, these are proto-chordates, hey? These are the more not so developed chordates. We spoke about some origin and what went wrong, and then we stopped here. And we spoke about, we're going to present the CEPHALOCHORDATA and the UROCHORDATA. Remember the three groups here? And the one group, the lancets, is cephalochordates. But remember the one group from last week is very similar to a cephalochordate, the Larvaceae. But the Larvaceae is actually one of these UROCHORDATA. UROCHORDATA means tailed. CEPHALOCHORDATA means head chordate. That's what we're going to look at today. Okay? Again, in terms of the subphylum, UROCHORDATA, CEPHALOCHORDATA, and vertebrates. Euro, the tail part, remember that. CEPHALOCHORDATA is the head part. Vertebrate, in between. Cephalochordates are lancets and amphioxus. So amphioxus is an old genus name. They're lancets today. They are laterally compressed. They are squashed from the sides to look like a fish. 3 to 7 centimeters long, translucent, which means you can see through them. They occur in different habitats, but we find them in the sea. The cephalization is the development of the head region, is a key feature that's moderately developed in amphioxus. They have a nerve cord that is not extended into the brain. It lacks paired sense organs of the vertebrates. Cephalization means increasing differentiation of sensory organs, nervous system, and the brain of the interior parts, the head side of the organism. Okay, the cephalochordates laterally mean head cord. That's what we've been speaking about already, head cord. CEPHALOCHORDATA. They resemble, and remember, until recently, we only thought the cephalochordates were the only protochordates, the more uncomplicated forms. Please, ma'am, you must move with that sheet, eh? You got to move it with that sheet. We got fossil records from 500 million years ago, according to our carbon dating theories, and Picaia. They're anatomically similar to amphioxus. Okay, branchiomata, branchiotoma. But they have all of the 5 chordate characteristics. Remember the 5 chordate characteristics. Here there is 6. Remember, I've been telling you that we used to include a 6th one, but the 5 ones are the dorsal and tubular nerve cord, pharyngeal slits, and the endostyle. Endostyle is the glands. You know, your thyroid gland or your mucus glands. Number 4 is the notochord. Yes. Number 5 is the post-anal tail. And remember, we used to consider the myomas, the chevron-shaped muscles. It has all those characteristics. In terms of the anatomy and the feeding, it moves water through the pharynx using cilia. That's its feeding characteristics. Is this the same thing? Yeah, water moves in. Water is taken in through its mouth, through the pharynx, where the cilia take out all of the food out of the water. Remember the mucus layers on the cilia. Cilia is like little fingers, the little hairs that occur within its pharynx. They take the food out. There's a mucus layer around the cilia. The mucus layer comes from the endostyle. And as the water moves through its pharynx, all of the little food particles, the phytoplankton in particular, is trapped in the mucus layer, and then it's absorbed into the organism that way. Okay. The hepatic cecum is a part of its, so the hepatic cells, when you start to get to see words like hepatic, we're starting to refer to the type of cells that are commonly a part of your liver. Okay, so there are different cells that are starting to form to allow different types of digestion in the organ. So they have a hepatic cecum, and then the water is then ejected out of its anus. Okay, the antipore. So the food is collected by all the cilia. The cilia is absorbed into its gut, the intestinal tract. The gut then has this hepatic cecum, which is part of the digestive process. And then whatever's left of the food is excreted out of its anus. But the water goes out of an antipore. Okay, the water comes through here and comes out of an antipore. And the antipore is where the antipore is like in modern fishes, you see that they absorb water through their mouth, through their gills, and out of their gills. The circulatory system is closed, which is similar to a fish. This is unique to this Amphioxus. Has no heart. Peristaltic contraction of the ventral aorta. Your peristalsis is the same type of movement that you use to move food down your esophagus. So it moves like this. Different parts of the tube squeeze, and it moves it along by squeezing different parts of the tube. So they use their muscle to squeeze and move their blood through its circulatory system. It doesn't have a heart. Pretty crazy, huh? It has a ventral aorta. It has a branchial arteries, and the branchial arteries are the arteries that goes through its pharynx, and that's how it's used to get some oxygen into its circulatory system. So it's actually similar to a fish in that sense. Paired dorsal atore, capillaries in the organs, veins, and a ventral aorta. Okay, so this is the very basic circulatory system of Amphioxus. I think what I'll do is I'll just improve the picture for you a little bit. It's quite cute, but I think I'll improve a little bit. But that's what it looks like. It only transportation of nutrients, not oxygen or CO2. So the circulatory system only transports nutrients. Okay, where did I go? Sorry. So its nervous system, it does have a hollow nerve cord. The ocells are simple, unpaired sense organs that function as a photoreceptor. So photoreceptor means that it is sensitive to light. Yeah. So it can see day and night. So it probably has dial activities. Dial means that during the day and the night, it has different behaviors. It does different things differently. It's active during the day and it's inactive during the night. And that's very useful if you're a little animal like this, because during the day, generally the temperatures are going to go up, and your opportunity to maximize on your metabolism and the availability of food goes up, and then at night you can slow down. Guys, please don't disrupt everyone. Okay, reproduction. Sexes are separate. Do you remember with the UROCHORDATA, they were hermaphroditic? Male and female organs in the same organism. But here you have separate males and separate females. The gonads produce the gametes, the sperm cells, and the ovums, or we refer to them when they're not very complicated as gametes, through the atrium into the antipore. External fertilization occurs in Amphioxus. Okay, so that means like a fish, they release their eggs, they release their sperm cells, and fertilization occurs outside the body. Additional features are similar to those in vertebrates. The hepatic cecum, which is the same as compared to your liver and your pancreas, closed circulatory system, and the myomers are the different types of muscles. So there are some important characteristics that Amphioxus have which are shared with other vertebrates. That's very important to remember. And remember, the myomers is that type of chevron-shaped muscles that occur. Okay, and then just refers to your next part to read. So guys, what's important that you need to know, let me just go back to the diagram. Okay, so we've spent some time on the prochordates, the protochordates. And what's important here is to understand that there's a relationship between these early protochordates and the rest of the vertebrates. So you've got UROCHORDATA, CEPHALOCHORDATA, and the VERTEBRATE. And all of the vertebrates together are all in this line here. So we've only discussed these three lines, the CEPHALOCHORDATA, UROCHORDATA, and VERTEBRATE. Those three, 1, 2, 3. You understand that? And now that we've discussed these two, and we've been able to talk to you about what are the characteristics of these two, where do you find them, what are their features, how does it relate to the other other cephalochordates, and the vertebrates. Now we move on to the vertebrates. Okay, so this is the last lecture we're going to have on the protochordata. Next time we're moving on to the fishes. So we start with the fishes. The hagfishes and the lampreys, they don't have any jaws, agnatha, remember? So that's what we're going to do next. Then we're going to move on to the bony fishes. And in between the bony fishes are these little chondrichthyes, which are the cartilage-focused or cartilage-based sharks. Okay, so we're going to work on the jawless fishes, the hagfishes and the lampreys. The sharks, which have no bones, they have only the hardest thing is their teeth. Then we move on to the bony fishes, and the rayed fishes and the lungfishes, okay, and the coelacanth. We're going to do some work here, and we're going to have some nice practice on this as well. Then we move on to the amphipoda, the amphibians, and then I'm going to do a little bit of the work on the reptiles. I'm just going to start it, but I think we're just going to initiate it. And then Dr. Minnie is going to take over and do mammals and birds with you. So for the rest of the semester, I'm focusing on the fish. The real stuff. Okay. One of your practicals, guys, listen. When I told you about the practicals on the toads, why are you going to do a practical on the toad? Because you need to understand the life cycle of the frog. What does the frog do? Amphibians. They lay their eggs in water, and then they go through a metamorphosis, and then they go and live on land. You understand that? That's a very useful idea to help us understand how it's possible to go from being dependent on water because of your gametes and your fertilization. The development of embryos occurs in water. So here, everything depends on the water itself. Then these guys also depend on water, but they have the ability to metamorphosize and go onto land. These guys have to carry the water with them. If you live on land, you need an integument to help you carry the water with you. You with me? And that was a major development. So that's why these guys are important. And what I want you to do for your prac is we're going to set up a tank with a bichana land, bichana manzi, and then the frogs are going to breed, and you guys are going to document what day, what happened. So by next week, I'm going to try and get the tank set up with some frogs inside, and your job for each of your groups on your own is to go and record. The frogs were in the tank on the Thursday, whatever. Then they were fine. They were being fed, they were feeding, there were no babies, they're just croaking and making noise and the breeding. Then they started pairing. And when they started pairing, then they started breeding. And they were producing these eggs. And then what happened with the eggs? The eggs hatched. And we had some tadpoles. And then the tadpoles started maturing. At each stage, guys, I'm going to have the adults in and the adults are going to breed. When the adults are bred and they're done, I'm going to take the adults out. Why? Because they'll eat the babies. Then you can focus on the tadpoles. Then you watch the tadpoles, you see what stage they're going through. Then they start to get legs. You understand? And you're going to do a project on that yourself. So in the textbook is the whole life cycle of the amphibian. You guys are going to do it. You're going to see it happen in real life. That whole cycle. Cool. I'm going to give you a prac for fish as well. And your prac for fish. Hang on. So conservation 102 is going to the field tomorrow. Ne? Does Con 102 have groups yet? Your groups are quite similar. Hey? Your class is similar. It's just the guys from last year who are different. So I'm going to try and apply your same groups to Con 102. It's actually 1, 3, 5, 7, 9 Con that's going to the field tomorrow. Sorry, guys. Zoo's prac was done. Next Monday, Zoo prac. We're going to decide whether half of you go to the field or we do pracs. Your one Zoo prac that you're going to do with live animals is the frog group. It's the frog prac. Because all of you are going to the field for Con and you're going to see fish anyway. So I'm going to give you a desktop fish prac as well for Zoo. So Zoo, your prac on fish is going to be desktop based. You're going to have an assignment to do. You're going to have to go through the assignment and answer the questions and try and figure out what is the requirements of different species of fish. For Then for zoo as well, you're going to be doing an amphibian prac. That's all you have to do for zoo, for pracs. That's it for zoo, done. For con, we're going into the field, you're going to split up into 2 groups, and you're going to go through the whole ecological health of the Niles River. Okay. Cool. Everybody happy?